Hello, my name is Ryan Bailey, and I'm pleased to be presenting to you today some recent work from our group in developing droplet microfluidic tools for uses in high-throughput biotechnology. I'd like to thank the organizers for the invitation. I'm at the University of Michigan in the Department of Chemistry and affiliated in the Department of Biomedical Engineering. So while we're really developing kind of core tools that can be applied to many applications in high-throughput biotechnology, what's motivated my group are actually applications in high throughput and low input epigenomic studies. So I'll provide a small amount of context as to why we want to develop these types of tools, which I hope demonstrates the utility that these can be used, of course, in many other applications as well. So I just want to point out that this work is being done with a team at my lab as well as several people in Tomas Ordog's group at the Mayo Clinic. So it's a little way of motivation. Chromatin's organized with these nucleosomes being the most basic unit. So we often think about DNA, there's a double helix or as a chromosome, but in between there are all these hierarchical structures of chromatin structure. And again, the core unit is this nucleosome, which is a protein complex. These are histone proteins, which have these small polypeptide tails. In addition to modifications to DNA, which can change the spacing or compaction of chromatin, these modifications to these polypeptide tails is also very important. And these work together with readers and writers in the cells, as well as transcription factors, to dynamically control gene regulation. This turns protein expression on and off effectively. Now, there are many needs to develop, I would say, clinically applicable precision epigenetic methods. So I think it comes as no surprise to most in the audience that epigenetic signatures or dynamic epigenetic regulation has profound roles in development, but it also underscores many other diseases, including cancer and others. Importantly, there's a growing pipeline of pharmaceuticals with epigenetic targeting modes of action. So what's really needed are these essentially companion diagnostic assays, but in the epigenetic space as opposed to the genetic or the protein biomarker space. So there's a real need for this. So there's really a need to understand functional consequences of gene-specific and genome-wide epigenomic lesions, and we think that this is going to be important in a new wave of personalized medicine. And to accomplish this, we really need fundamentally new measurement tools. There are pretty good tools that are used in, say, academic and medical research labs, but these really do not have the scalability and throughput needed to robustly establish clinical significance by looking at large patient cohorts and things of that sort. There are several different classes of epigenetic assays. You have potential for translational significance. Many of the tools we're developing can be applied to many of these. One that we're not focusing on in particular is looking at DNA and RNA modification. So things like methylation and hydroxymethylation of cytosine are handled right now quite well by bisulfite sequencing. There are assays that are general probes of chromatin condensation. That includes things like MNA-seq, which I'll talk a little bit about later, DNA-seq and ATAC-seq in particular, which has been used quite a bit recently. And then finally, there are these antibody-based recognition assays where you're looking at specific histone marks and the association of those marks with particular genes. And the most common version of this is chromatin immunoprecipitation, or ChIP-seq for short. And that's what we've really been focused on for a long-term goal. And really it's because from a kind of engineering microfluidic standpoint, it's the assay that has the most individual steps, and it's theoretically that would be the hardest to automate, yet have the largest impact if we were able to automate this in a completely microfluidic format. Just want to go over quickly what the ChIP-seq workflow is in bulk and kind of what the results look like, kind of again as framing this talk a little bit. So you typically start with a cell sample. This could be a culture cell sample, a patient sample, something like that. You may want to cross-link if you're looking at, say, weak binders like transcription factors and their interaction with DNA. But that's not always necessary. What is necessary is you always need to lyse the cell and digest the chromatin. So we're going to try to cut it down to these mononucleosomes, the smallest fundamental unit of chromatin structure. You then throw in magnetic beads that have antibody on them. These antibodies have specificity for particular modifications on the peptide tails, shown here these red triangles. Then you can use the magnetic nature of these to pull out just the mononucleosomes that have histones having a certain mark. You then can release that DNA, purify it, and take it downstream to something like sequencing. And here's an example on the right from our collaborators at Mayo Clinic, just showing some ChIP-seq data on a gastrointestinal stromal tumor GIST sample. And here they're looking at 3 different marks: a promoting mark, H3K4 trimethyl, and 2 different repressing marks. And really just the only thing that's important to see is we see these very defined peaks for these promoting marks and kind of orthogonal islands or lack of marks, lack of reads in regions where overlapping between promoting and repressing marks. So that's kind of the long-term objective. So what are the impediments to that, to the clinical implementation of that general scheme I showed you? Well, while these methods are very well established, they're laborious and highly influenced by the level of user expertise. Even in an academic lab, in my lab, it can take someone a couple of years how to really robustly run these ChIP-seq experiments, the IP pull down. So this makes them very much not amenable for large-scale studies or discovery of biomarkers, epigenetic biomarkers. Also not suitable for use in central laboratory settings, just requires too much technician time. Another potential limitation is that they have relatively large input requirements because of the many steps involved. This means you cannot apply these techniques easily to sample-limited types of samples, such as circulating tumor cells, tumor biopsies, or more specifically laser capture microdissection to look at heterogeneity, or cancer stem cells. So there's really an opportunity and a need to automate to go to lower input and much more high throughput for these assays. So the idea would be that rather than a 4+ day assay that requires millions, 10 millions of cells, you're looking at 1 mark at a time. It's very user independent. The ideal would be using a microfluidic device to push this process down to hours, taking advantage of enhanced mixing and diffusional transport that happens on these short micrometer link scales. Push this down to hundreds or thousands of cells, maybe single cells eventually. Make this parallel so we can look at multiple marks or multiple transcription factors simultaneously. Really a huge benefit would come from being able to automate this using microfluidic tools. So we're not the first ones to think about automating or using microfluidics in applied to ChIP. These are several different notable examples from literature, but all of them really still have some limitations that I just wanted to point out because I think informs why we're trying to use the approach that we are. One is that while microfluidics are fantastic at going to small volumes, they end up having gigantic surface to volume ratios. And we can think about any surface at a microfluidic device potentially as a place where we could lose sample. Sample could stick onto the walls of the plastic or glass that the chip is made out of. Many of them often have very complicated valve controls. You have to be opening and closing different valves to control what's added or taken out of a sample. This requires skilled operation. There's often sample loss between these steps, so many of these involve, like, say, filling of loops and things like that. And if you want to add reagent to a loop, you have to push out a little bit of what was already in there, so that gives you sample loss. Other of these approaches are very much optimized for limited or fixed volume samples. And that means if you have a sample that's larger or smaller than what you have optimized your device for, you have to kind of go back to the drawing board and change geometries or antibody loading or things of that sort. Finally, none of these approaches have been able to fully integrate all steps. So you can take just a cellular input, and from that cell input, prepare the nucleosomes on chip and do the immunocapture on chip. Usually they'll start with, say, nucleosomes that have already been made. And our goal is to take cells for inputs and eventually have sequencing libraries on the backside. That we're not quite there, so I'm not going to be able to tell you about that part today. And then finally, they have not side-by-side validated with conventional chip. And this is very important in a translational, from a translational perspective, because clinicians are not going to believe or readily believe the results from a microfluidic device. They cannot be compared side-by-side with bulk scale preparation. So one of the approaches that we've taken is to be able to develop devices that can be upscale. We can run them for longer periods of time and actually accommodate the large sample input that's typically used in a bulk chip experiment, so we can do just robust side-by-side comparisons. So our approach is a little different than the others I talked about before, is to use droplet microfluidics. And these are segmented liquid-liquid flows in which we have a continuous oil phase. This is often a fluorinated oil. Think about it like a Teflon that actually makes it so that the sidewalls of the channels aren't sticky, which is important because we don't want to lose material binding to the sidewalls. But we can inject a aqueous liquid into this, and it will branch off and actually form a small droplet, break off into the flow. So we make these continuous flow of droplets. So as I mentioned, there's this really nice advantage because we have limited non-specific binding. Also, each one of these is essentially its own little reaction chamber, which means we have very limited cross-contamination from droplet to droplet or potentially sample to sample. We don't require additional mixing elements in these types of devices because there's this natural convective flow inside of droplets that leads to very nice mixing internally within the droplets. So that's an advantage. And also, as I mentioned before, these have indeterminate operation. I can run this device for a short period of time and make just a few droplets, or I can run it for a longer period of time and make, thousands of droplets, even just in a couple seconds. So that allows us to think about how we can scale this to accommodate different types of sample input. So here's kind of our strategy, if you will, to move chip to a chip. So I've kind of shown, again, the different steps involved with bulk chip, from taking the cells to isolating nuclear chromatin, digesting the chromatin, then followed by the immunoprecipitation and DNA purification. So we've actually broken this into eventually 3 modules, but the 2 that we've really focused on first, and that is cell encapsulation. And we're going to do this with the presence of detergent and microcalcal nuclease just to start that cell lysis digestion immediately. This incubation of our cells for fixed periods of time, for defined periods of time. And then at the end, we can actually add in our EDTA buffer, which chelates the calcium out of this enzyme we're going to use, and it quenches the reaction. And at the same time, we can add in magnetic beads if we'd like for the downstream immunocapture process. We then have developed a few different approaches towards magnetic bead washing, and I'll come back to this, but we use what we call fondly a car wash device, and I'll talk about what this is, to do bead washing as well. And then finally, what we haven't brought on chip is this DNA purification and downstream analysis. There's precedent for doing these things on device as well. So as I mentioned before, we're going to be using an enzyme to digest our chromatin. In large academic labs or research labs, people typically use or can often use sonicators, which are actually very expensive and difficult to operate, very sample size dependent. One of the advantages of using this microcalcal endonuclease or emanase is that it's very gentle. It doesn't degrade any proteins, doesn't require any flexible or specialized equipment, flexible reaction volume. We can control the incubation time very precisely in microfluidic devices. So this is an endonuclease that will basically just chew up any DNA until it gets to a nucleosome, at which point it can't eat anymore. So this is the steps here. We're going to combine our cells with our detergent and our digestion buffer, make droplets. We can incubate them in these delayed channels, then we can quench the reaction. So here's an example of what one of these devices looks like. Again, up front, we have where we make the droplet formation. In this region here, these are where we do the incubation. And it's really important to point out that when we make these microfluidic devices, which is actually quite simple, we can change the number of these delayed channels we have. So I'll show you some data where we just actually stop after 1 row of these delayed channels for a very short incubation time. But that's something we can control very nicely, very easily on chip. And then finally, this injection down here, this quenching buffer and droplet collection before moving on to the next module. So here's just a kind of side-on 3D rendering of this, and I wanted to show a few videos so you can get an appreciation for what these look like in action. Here at the droplet formation port, we have our cells come in. You can see these little cells floating down the channel. And this other channel that's merging with it is an aqueous buffer that has the detergent, the microcalcal nuclease. This will then form droplets, and many of these droplets are actually empty, but if they have a cell, just statistically based upon Poisson statistics, they'll only have a single cell. So we're effectively doing single cell processing, but we're just doing it on many thousands of cells simultaneously, but it's important that we're doing single cell processing. Here's the beginning of this delay channel. So we made the delay channel a little thicker or deeper so we can accommodate more droplets, but here you see these droplets coming in. Here's a droplet per cell limit entering the delay channel. At the end of the delayed channel, again, we can have as many of these as we want. We see the droplets are still intact. So they just move through these delay structures to get nice homogeneous incubation time. And then finally, we're able to inject this EDTA-containing buffer, which chelates the calcium and stops the enzymatic digestion at the end. So here's a picture of the microfluidic device filled with a food dye, so it's easy to see. You can see it's about 5 centimeters by, say, 3 centimeters in size, so quite small, quite compact. And here's the result if we do this micrococcal nucleus digestion using just one row of delay channels. So we know that we're doing a very short incubation here, only about 45 seconds. But what we see is just after 45 seconds of incubation, we can already clearly see mononucleosomes, dinucleosomes, trinucleosomes, and larger, relatively undigested peaks of chromatin. But again, we can control the number of channels that we have, delay channels, to optimize our incubation time, and that's shown here. These are electropherograms looking at samples from 45 seconds up to 305 seconds. We can see this nice large peak of mononucleosomes really in all the samples, but at 45 seconds we still have some di and trinucleosomes. They really go away so we get a longer incubation period. This is actually slight over-digestion of the DNA, which is actually really nice for the nucleosome positioning assays that I'm going to talk about later. Showing quantitatively here, we see that as we go from 45 even just up to 90 seconds of incubation, we already have on the order of 80% mononucleosomes, which is again what we desire for these nucleosome positioning assays. And if anything, we see that our on-chip here in red performs, in some cases even better, or excuse me, the on-chip on black performs even better than the off-chip done just in a tube. So really nice processing of this chromatin to mononucleosomes from whole cell input using this device. Also, since we're really processing these cells one at a time, it doesn't matter how many we process. So we can see as we go from 125,000 cells down to just 2,500 cells, that we have essentially equivalent mononucleosome yield. So again, that's a benefit of doing essentially, many one after the other single cell processes. And we can go lower, but actually we don't get enough signal out of our electropherograms to do this. This is actually one of the limitations as you start to go to single cell omics, is you don't have some of the same characterization methods available. It looks like this method would be amenable to single cell processing. And again, also, since we see consistent results, this allows us to upscale. We can run this, and we can look at 10 million cells just by running this device for a longer period of time, which we think is going to be important. So here's some MNA-seq data that I'm going to show. I know there's a lot of data up here on the slide, but we're going to focus on 4 gene regions. The first 2 I want to focus on are here on the left, EIF1 and fumarate hydratase. These are both genes that are constitutively active in the Jurkat cells we're working with. So we see here flanking the transcription sites. These genes are actively poised for expression under active regulation. So we see these nucleosome-free regions in all the samples, whether they're on-chip or off-chip, whether they're free, which means we've made the sequencing libraries without PCR, or whether we've used PCR to make libraries. We see consistent nucleosome-free regions flanking the transcription start site, which is what you'd expect, because you need to have a free region for the transcription machinery to come down and recognize that this is where you're supposed to start transcribing and binding and processing. In contrast, when we look at these 2 other genes, GFAP and tyrosine hydroxylase, these are both developmentally repressed in the T cells. Again, these are from Jurkat cells. So we see here with the transcription start sites, no nucleosome-free region. So this is consistent with things that are developmentally turned off. If we look genome-wide at all of the analysis of genes under active regulation, we can see really quite remarkable indexing from plus and minus 1.5 kilobase pairs from the transcription start site. Again, on-chip, off-chip, with and without PCR, we see really fantastic agreement, 96% overlap in nucleosome position between any pair, regardless of using PCR, on-chip or off-chip. So this is really quite very good processing. Also, if we actually look at quantitating the number of nucleosomes per cell, you can kind of go through the math and you can see that there should be, given the number of base pairs per cell and how big a nucleosome is, roughly about 1.5 times 7 nucleosomes per cell. And again, we hit that right on right on the head. In fact, we don't see the PCR bias, which suggests again we can go to even smaller input in the future. So I'm going to revisit that chip workflow. What I've talked to you about before is this first step, which had never really been done before, but making high quality chromatin in a microfluidic device. And now move to this immunoprecipitation step, where we're going to be adding in magnetic beads and be able to control those beads to pull them out when they've bound to their target species. And so we've been developing for several years in my lab a range of different components that can potentially enable immunocapture. And I'll talk to you first about one of the ones that we developed earliest, which is what we call the K channel, because if you turn this device on its side, it looks like a K right here. And in this case, we have a counterflowing fluid, and what we find is that if we have a higher pressure in this counterflow compared to the droplet flow here, and we have an electric field present. This is an electrode right here, just a saltwater electrode. It destabilizes that interface, and we can inject fluid into this droplet. Using the exact same device, we can reverse the pressure. So now we have a lower pressure from the counterflow, and now we're pulling off sample. So the same device, we can both add and remove fluid in a reversible and highly tunable way. At moderate pressures, especially if we turn off the electric field, we see no fluid exchange at all. So this could be, we could turn off the module if we wanted. And then finally here I'm showing it with a oil counterflow, where now we're splitting the droplets in half. So these are a range of different functionalities using just this one K-channel tool. And we like this because though we're chemists, I've taught a lot of chemical engineers in the past, and we like to think about things sometimes as unit operations. So here we have these dynamically tunable unit ops that we can use for microfluidic samples. One other thing I should mention, I didn't mention it earlier, is again, the droplet generation rate we typically work at is at hundreds of hertz. So these droplets are passing by this interface extremely fast. You can see the time on the videos here on the top left corner. But we're making these droplets very quickly, and the reproducibility of this process is really quite phenomenal. Again, kind of getting back to this idea of unit ops, here's just an example of how we can start to think about doing immunoprecipitation by pulling off magnetic beads and decanting a supernatant. Upstream here, we've made droplets that have magnetic beads. We then flow them across this K channel that's poised to do droplet separation. It's a little hard to see, but at the very bottom of this channel, you can see a few dark splotches. These are beads that are being pulled down towards a magnet, which is just out of view. And then if we look downstream at this decanted supernatant and these droplet-containing beads, we see really nice bead enrichment, 96% bead enrichment. And again, we're doing this at hundreds of droplets per second. These droplets are on the order of about 150 picoliters for reference. So actually very nice size for kind of single-cell analysis. And again, just kind of showing this unit operation. Here we're splitting a droplet, we're respacing them, and here maybe we're adding in a rinse buffer. It's the type of immunoprecipitation you might want to do. So we can really put these together and operate many of them side by side, or in sequence, I should say, and do quite complex operations in this way. Now, it turns out that trying to still do 4 buffer exchanges and buffer washes with very high fidelity is still difficult with that module. So Steve Doonan here in my group, who's now a postdoc at Washington University, came up with this really clever approach, which he named very cutely the droplet car wash. And car wash stands for coalesce, attract, resegment washing. And this is just showing how this works schematically here. We have a magnet at the bottom of the device. We have our droplets containing magnetic beads. They come in. We're applying an electric field across this interface. This is an electrode. We bias this aqueous flow back here at the input. These droplets will then coalesce. And once they coalesce, the droplets are free to actually be pulled magnetophoretically towards the bottom of the channel while the fluid is flowing left to right. So when we do this, we actually can get rid of all of the excess material in the droplet that's directed just out to waste. We can then collect just the magnetic beads and then resuspend them with an oil flow at the bottom to do subsequent downstream processing. So this is an example of the car wash module in action. So here are the droplets, some of them containing beads coming in, and we can see that they fly across down towards the bottom here because they're magnetophoretically pulled to the bottom of the channel. And all the excess material up here just goes to waste. we can show this even more clearly. So our bead recovery is quite good, about 98%. But if we spike the incoming droplets with fluorescein, a fluorescent tag, and do microscopy on this, we can see that all the fluorescence really stays up here at the very top of the channel. None of it makes its way down, or very little of it makes its way down to where the droplets, the beads are being re-encapsulated. And here's just a cross-sectional fluorescence image with the 1% standard here for reference. So we're doing at least 100-fold dilution of the excess material, or essentially washing the bead by doing this, and it's quite efficient. Now, in the chip process, there are multiple buffers you typically want to use. You want to use 4 buffers, typically a low salt, a high salt, a lithium chloride, and what's called a T buffer. So taking advantage of one of the properties of microfluidics, which is the fluids don't like to mix that well if they're flowing side by side, we can actually create a multi-phase flow. So in this case, we labeled, our bead sample is unlabeled, has the streptavidin beads. Our low salt buffer has fluorescein. Our high salt buffer has no fluorescent label. And the lithium chloride buffer has fluorescein again. So these are actually co-flowing in the bright field image. You can't see anything, but if we wait just a second until it turns fluorescent again, you'll be able to see, and you see the beads traveling through. Oh, here comes a bead. They come down, and you see the fluorescence is all confined up here. We have a little bit of leakage in this early design, but this is suggesting that we can do 4-phase flow simultaneously. And here's we got a fluorescence cross-section. So we're actually pulling through and removing all this excess material that this is what we want. We aren't pulling now. We want to get rid of in the immunocapture experiment. So here we're doing this about 70 hertz. We've slowed it down a little bit, then resuspending droplets into the beads into new droplets. So there's really a lack of a bulk analog for how you do kind of these multi-phase washes simultaneously. So this is something we thought was pretty interesting. Finally, or just to kind of put this into context, I've described already upstream how we can use the emanase device to digest chromatin. We can use this car wash to wash the beads. And at present, we're just doing our DNA purification and library preparation off chip. That's something that we're actively working on bringing on chip as well. And again, there's some precedent for doing that already. But I just want to share with you one example. We don't have quite the finished story here, but this is one example where we're looking at manual processing and on-device processing of looking at 3 different genes that are pulled down through this H3K4 trimethyl. So this is histone 3. It's a trimethylated lysine 4. That's the nomenclature for that histone mark. And we can see an enrichment of this BRG1 that's specifically associated with this histone mark. And these other 2 genes are off-target controlled. We see they're not enriched. And this is a log-log plot. So greater than 2.2, suggesting at least a 10-fold enrichment. So we think this is really promising, and we're moving this to other marks, and we think that has good opportunity. So just as a final thing, I wanted to share with you a different tool that we've been developing for droplet operations. So in the micrococcal nuclease device, we incubated for a total of 305 seconds, or about 5 minutes. And while that works very well for micrococcal nuclease, there are certainly some assays where we want to do much longer processing steps. So, this might need tens of minutes or more of incubation within droplets. And space is time in a microfluidic device. So if you want more time, you need the droplets to travel slower or for a longer distance. And when you do either one of those processes, what you tend to generate is back pressure in the device. And this can have lots of different problems. And another consequence is that if you remove back pressure by just making your channel really big, then your droplets, they start to diffuse, and you get a lot of temporal heterogeneity because they diffuse within laterally or from front to back in time, if you will, and you get more temporal heterogeneity. So what we've been interested in is developing a fusion of on-chip droplet processing and off-chip incubation, so we can take advantage of all the great things on-chip, but then actually I'll give you the word here. It's a little teaser. Pack the droplets well. If we tightly pack them, we can get really nice on-chip incubation with a much wider diameter external tubing that can work for this. So again, I kind of spoiled my surprise here. Most of the time, people use droplet fluidics with oil fractions on the order of about 50%. That means you've got about half the volumes oil, half the volumes droplets. They're kind of one-to-one spacing periodically. and this leads to significant dispersion of these droplets. So we're interested in removing this excess oil after we've done all the processing, so we can pack them and then put them into this on-chip incubation device. And we just published this earlier this year. But Ming Sun, a postdoc in my group, came up with a really clever way to do this. So we've made droplets up here, upstream, and once you make droplets, they come and they pass an oil removal module. And so what Ming did is he actually punched a hole in the device. These are all PDMS devices, polymethylsiloxane. And at the bottom of this, he actually puts a PTFE membrane. And remember that I mentioned that the oil we use is typically a fluorinated oil. So that fluorinated oil actually likes to interact with the PTFE better than water does. So if we apply a slight negative pressure to this oil draining module, we can effectively pull the oil out. And you can see right here that we take dispersed droplets at roughly 1-to-1 oil to water ratio, and we can selectively pull out all the oil. So we can densely pack these droplets. These are actually posts, these white circles, in the incubation channels to give it some stability so it won't collapse. We can really tightly pack these droplets. And what we find is that we pack the droplets really tightly. So here's just showing a device, kind of two devices put together. So we make droplets, we pack them, and then we actually have this bridge incubation tubing. We actually take the chips, the droplets off chip in this much wider diameter tubing before we bring it back in to another module here that can respace the droplets for downstream analysis. And by just using the longer pieces of tubing or wider diameter tubing, we see that we can get really nice homogeneous incubation up to 20 minutes. But we could actually, there's no reason we shouldn't be able to go longer. We're using a fluorescent readout using an enzymatic substrate, a pair that can turn over and create a fluorogenic substrate. But here we can get 3-minute incubation, 6, 10, and 20. We can actually see an increase in the product, the enzymatic fluorescent product that's generated before it just starts to be depleted, and that's why it plateaus out. So we think this is kind of a clever way to kind of get at this world-to-chip issue of how you can do a lot of processes that work well in a microfluidic device, and then you can get much longer incubation for kind of all the bulk scale assays that many people, or analogous to the bulk scale assays that many processes in screening and things like that would want, and then we can put them back into a device whenever we'd like to, control that incubation timing and do subsequent processing. So we think this is a kind of a clever little approach to doing this. So I just want to conclude quickly here. Droplet microfluidic modules can be created that are tunable, provide unit operation capabilities on the hundreds of picoliters as we typically work on to the nanoliter volume scale. You can go down to femtoliters with most of these. You could go up to microliters if you really wanted to, but nanoliters and picoliters is kind of the sweet spot of the analyses we are interested in. In general, we think droplet microfluidics presents an attractive approach to epigenomic studies because of these dynamic input capabilities, this lack of non-specific binding. We have very simple device design, so none of our devices we ever have any active valves. We're not opening and closing channels dynamically. We might change the pressure on a K channel to make it, inject rather than remove, but these are, in the grand scheme of things, quite simple microfluidic devices, very passive devices. I demonstrated that we were able to show efficient chromatin digestion for the first time in a microfluidic device that has outstanding performance relative to bulk methods and allows us to go to lower input requirements, potentially even down to single cell processing eventually. We're still working on chip. We think we're pretty close to getting it all working, but I showed you that immunoprecipitation, that car wash module that was really important for a lot of solid phase extraction approaches that might only be done in droplet microfluidics. And then finally, I showed just a little teaser there at the end, this recent, kind of world-to-chip interface where we use that PTFE membrane to allow us to very tightly pack droplets, but yet keep them stable. And when we tightly pack them, we can use an off-chip incubation for longer periods of time without because once they're packed, the droplets can't diffuse past one another. So you don't really lose the time indexing of droplets when you pack them. You lose a little bit, but not very much compared to if you had these just floating in a tube with 50% oil. They're present. So with that, I'd like to again thank the organizers for the great opportunity to speak to you today. I want to point out Ishu, Steve June, and Gloria Diaz, who've done most of the work in my lab on this, funding sources, my collaborators at Mayo Clinic. And again, I'd be Really happy to have been invited. It's a great pleasure. And I'm happy to take any questions that you submit. I think I'll get them via email, and I will try to get back to you as soon as possible. I hope you learned something today. Thank you very much for your attention.